Can everyone see this? I'm going to put this on slideshow.
Yes, we can see it.
Okay, so I'm going to talk today. First of all, I'm Richard Crist. I work over at the CMB in the TRL building. And like I said, geneticist by trade, but over the last year or so, we've been working on single-cell transcriptomics in schizophrenia. And I'm going to talk a little bit about methods, because this is a pretty new method, a little bit about what this gets us over, above and beyond bulk lysate, and then I'll talk about some of the early findings we had here, because we're still analyzing the data. No? There we go. So schizophrenia, I'm sure most people are aware here, psychotic disorder, chronic, affects about 1% of the population and has really devastating costs on individuals and also just society at large. And like with most disorders, we've already looked at bulk transcriptomics. In this case, most of the stuff's either by microarray or RNA sequencing. And there's been a large number of studies across multiple regions, cortical and subcortical, found plenty of differentially expressed genes. Some of those have been replicated, a lot of the stuff not so much. Some of that's due to small sample sizes. Some of that's due to different brain regions, and so it's hard to really parse what all this means. But people have done pathway analysis. They've done gene ontology, enrichment stuff, and they found things that make sense. So neural development pathways, stuff related to axon guidance, and stuff related to inflammation and immune-related proteins, which makes a lot of sense given the genetic results from the schizophrenia GWAS. And the really big one here in terms of bulk lysate RNA-seq data is the Common Mind Consortium. And you can see here it's got 258 cases, 279 controls, and this was from the dorsolateral prefrontal cortex. And they found 700 differentially expressed genes. It worked out surprisingly well here in terms of equal numbers of genes upregulated and downregulated, and we can talk about that a little bit later in regards to our data. Because ours was a little different. And I think it's important to note that this was actually a pretty diverse sample, all things considered. It's about 80% European ancestry, but there's a large amount of African Americans in here too. So pretty diverse when most of the studies so far haven't been. So why do we care about bulk lysate versus single-cell RNA-seq, which is what I'm going to talk about? And so the biggest problem here is what you see on the right, which is just a cartoon of the different cells that you find in neural tissue. And so the problem is that brain isn't made of a single cell type. Brain is made of multiple cell types, neurons, astrocytes, microglia, et cetera. And so even within those cell populations, you're seeing other distinct subpopulations that people have known for years, parvalbumin positive interneurons, different layers, layer markers, so there's different pyramidal cells at different layers that could have different expression. And when you lump them all together, you lose a lot of resolution on everything. So if you see something that's differentially expressed, you don't know what cell types it's relevant to necessarily. It could be relevant to all cell types. It could only be differentially expressed in some major subset. And on the other side, you might not be able to see genes that are differentially expressed in less common cell types. So if you had a cell that was 1% of the population, you're probably going to lose that in the mix, and you just won't see what is potentially a relevant differential expression event. And someone actually went forward and kind of proved this point in schizophrenia. So a few years ago, a group published two papers, one in 2017, one in 2018, and they used immunofluorescence and laser capture microdissection. And in these two papers, they pulled out, in the one, layer 3 and layer 5 pyramidal neurons, and in the other one, parvalbumin positive interneurons. And one of the things about laser capture microdissection is it's pretty time intensive in this case. So they pulled out 100 cells per layer for each sample in the first one, and then 100 cells in just the parvalbumin positive interneurons in the second one. But they had a pretty large case control population. And after running the microarrays, 1,400 genes differentially expressed in these pyramidal neurons and almost 900 differentially expressed in the parvalbumin positive interneurons. And the real takeaway here is that a lot of these genes were not seen in the Common Mind Consortium data or other previous bulk lysate analyses, which you kind of expect because these don't make up the vast majority of cells, and so they would have been washed out. You wouldn't have seen particularly small differential expression events in either of these cell types as part of the whole bulk lysate. So when you're thinking about single cells, I think it's important to look at some of the technical hurdles that pop up. And so clearly those last two papers used laser capture microdissection. Like I said, it's pretty low throughput. And beyond the low throughput, which just makes it time intensive, it has to be targeted. So they use staining to get their populations. But what if there's more than one population of parvalbumin positive interneurons? That might be interesting. You'll never catch that. Because you've now pooled them together. Moreover, we know that parvalbumin positive interneurons are relevant to schizophrenia, or we believe them to be so, and that's why they looked at them. But what about other cell types you don't necessarily know are relevant? So this goes back to sort of candidate gene study versus GWAS and genetics, looking for hypothesis-driven stuff versus casting a wider net and looking for hypothesis-generating experiments. Like I said, pooling cells can cause some issues, particularly with collapsing these subpopulations and just not really knowing they're there. We certainly know there's more than one subpopulation of layer 5 excitatory neurons that they would have lumped together in their study. And then finally, one of the big technical hurdles and sort of the lead into single nuclei RNA-seq, which is really what I'm going to talk about, is that frozen human brain tissue is the form that most of our brain tissue takes the form of, and the freeze-thaw cycle just ruptures cell membranes. So you don't end up with single cells, you end up with single nuclei in a soup that's left over from all the broken down single cells. So this is in contrast to, say, a mouse model where you can get fresh tissue and just dissociate single cells. Now, the good news is that people have actually looked into how correlated single cell and single nuclei transcriptomics are. And the answer is they're pretty correlated. You're going to end up with lower levels of transcript overall in the nucleus compared to the whole cell, which is not that shocking. But the same relative levels were found for 98% of transcripts in this one paper. Just worth noting, those other 2% may very well be very relevant to schizophrenia. I'm not saying they're not, but by and large, given sort of the difficulties in getting single cells, I think nuclei will get us most of the way there, and then we'll have to deal with those other issues later. So single nuclei RNA-seq in human postmortem brain really kicks off in 2015 with this paper from Blue Lake that was in Science. This is more of a methods paper, and they were working on this Fluidigm C1 platform that you see on the right. And basically, this microfluidics platform, you put your cell lysate or your cells in there in suspension, and they get caught in this tip that looks like the tip of a pipette. And there you can barcode each individual cell separately. But while it was originally designed for that, the idea that Blue Lake and company had was that they could use this for single nuclei. And so they fact sorted for neuronal nuclei, and they ran them through the system. And like I said, this is a methods paper, so it's a single brain, multiple regions, multiple different variations on the main method here, because they were trying to nail down how best to get this to work. But at the end of the day, what you got was this on the right, which is that based on single nuclei transcriptomics, they were in fact able to cluster out a very large number of different neuronal subpopulations. So this includes the big split here is excitatory and inhibitory neurons, as you'd be hoping to see in these kind of data. And then you see distinct interneuron subpopulations. You see regional differences. So these two around break E here, excitatory 2 and 3, are actually layer 4 neurons, but they're from different parts of the brain. So overall, this was a huge success, and what this showed people is that there was a way forward in human postmortem brain where we could look at individual cell types using transcriptomics from single nuclei, even though we couldn't get to single cell level. And so there were some issues with the Fluidigm C1. Most notable is, again, throughput. Over time, the C1 platform now I think takes 500, maybe even more than that, cells or nuclei at a time. But again, at the time it was 96, and so when they did 4,000 nuclei for that Blue Lake paper, that was a lot of chips they were running, and so it was incredibly expensive and incredibly time intensive for them. And so into the void of looking for high throughput stuff, this is the method that we're going to end up using, is the 10x Genomics Chromium controller. And so I'm going to just give a brief explanation of how this works, because I think it's important to understand how the entire system works, because this is probably for the foreseeable future what people are going to be using for single nuclei RNA-seq protocols. So you've got this chip that goes into the Chromium controller, and it's got 8 columns on it of wells, so you can do 8 samples in one run. In the first well here, you put in oil, which is just going to create droplets in the microfluidics platform later. You've got your cells and or nuclei along with the reagents for reverse transcription. And then you've got beads, and these gel beads are coated in oligos. The oligos, which you can see over here in part C, have this oligo DT region, but also each one of them has a unique molecular identifier, so that transcript will be uniquely barcoded. And then all the ones on the single gel bead also share an index, so you're indexing everything from a single nucleus. And then in part B here, everything runs to the microfluidic platform. You get these oil droplets, and you get out what are called gems, which are gel beads in emulsion. And what you're looking for are these ones that have one gel bead, which are the colored circles, and then the white circles in there, which are nuclei in our case. And so you want to have one gel bead, or one nucleus, and then all of the transcription reagents. After that, it's basically just reverse transcription. In our case, again, we're doing 3' bias here, so it's oligo DT, so it's amplifying poly A mRNA. And at the end of this, you add on your read 1 and read 2 things for Illumina, and you're good to go. You've got your library for sequencing. And I think it's important to note that there are pluses and minuses to this whole system. Certainly, the throughput is the big one. In our case, we're putting in a suspension that has 20,000 nuclei for each person, and we're getting out about 10 to 12,000 nuclei by the end. So that's much higher than you get at a much faster rate than you would get stuff for, say, Fluidigm or laser capture. You're indexing at 3 levels, so you're indexing at the level of the person, the nucleus, and the transcript, which the transcript part comes in handy for QC later. And then theoretically, it performs equally well on cells and nuclei, and it's theoretically cell type agnostic, and we'll talk about that at the very end of the talk, and why maybe it's not. The cons here are basically it's still very expensive, certainly, and there's fixed cost involved. So one of the other papers that worked on this, the depression paper, they captured really only 2,500 nuclei, and we captured 10,000. That capture cost was exactly the same regardless, but there's problems downstream for that. If you capture more nuclei, you have to sequence more to get the same amount of coverage per nucleus. So there's a lot of expense with this whole thing. Splicing isn't really addressed, which may or may not be relevant depending on your phenotype, but certainly appears relevant to schizophrenia in some regard. And then again, maybe not cell type agnostic in reality. So until this, until this last paper on Huntington's was published, there was really just 4. This was the big 4 for this field. So starting in the middle of 2019, there were 4 papers published: Alzheimer's, autism, multiple sclerosis, and major depressive disorder. And those have been very high impact. I think this is 2 Nature, 1 Science, and a Nature Neuroscience paper. And so people are very, very interested in what's been going on with this. The Huntington's one only has 2 people, where the other ones have like 25 to 48 people. So it's not really on the same level. But the important thing to note here is, despite the difference in disorders, they all found separate or differentially expressed genes based on case status. And they were all able to distinguish a lot of different cell populations, although we'll talk a little bit later about how many clusters you're expecting. They obviously didn't find the same number of clusters, and neither did we. So, So that brings us to the experimental design here. So we did 32 postmortem dorsolateral prefrontal cortex sections, 16 cases and 16 controls. This was a sample of convenience of what we had in the lab for another study. So unfortunately, it's mostly male instead of being equally split. Everyone here is European ancestry. What we're doing is basically what's shown on the right here, which is a modified version of what the NAGI protocol was. They did the depression paper that was previously published. And our stuff was modified by Ben Reiner, who's the other half of this work. And so the tissue samples were homogenized, washed, filtered, and then once they're clean, they're resuspended to send to the library build part. We used the Center for Applied Genomics. They did great stuff, so highly recommended on this. They have a lot of people who worked on the beta testing for 10x, so they have some technical knowledge that you might not be able to get elsewhere. But they did the library prep and the sequencing on their NovaSeq 6000. So I think it's important to note that there's a lot of quality control issues, and how you do quality control will probably determine a lot of what happens downstream, particularly for these data sets. And again, we'll talk about that a little bit later when it comes to whether this is cell type agnostic, because we set some of these cutoffs based on previous work and based on what's been known to give reasonable results, but there's a question of whether those are the correct cutoffs long term for people who are using 10x. But everything was deconvoluted through this pipeline with the 10x cell ranger. It was aligned to the pre-mRNA transcriptome for HG38, so the most recent version of the human genome. And then we cut out a number of things, genes that weren't found in a large number of nuclei. They're basically irrelevant to us. Nuclei with low numbers of genes or low numbers of UMIs, and again, those are individual transcripts. So these are just not a lot of information about these nuclei. And then we also cut out high UMI nuclei, and that's basically because high UMI, particularly at the very, very highest end, so in our case about like 0.5 to 1%, they appear to be doublets or multiplets, so there are multiple nuclei that have been captured in one library, and so again, they're not very informative. Using, again, a modified version of the NAGI protocol, we actually use highly variable genes, in this case a little over 2,200, to calculate our principal components. And then we did 2 runs on the clustering here. So the low resolution run basically gave us 2 useful pieces of information. One, we had low UMI clusters still remaining despite the cutoffs, so those were removed. And 2, we actually had to get rid of 2 schizophrenia samples, unfortunately. One of them clustered in a single cluster by itself, so very probably low quality. A second one clustered by themselves in 4 or 5 separate clusters. So I'm not 100% sure if that's low quality or if there's something where that person actually just happens to be an outlier in the population and they just weren't clustering with the rest of the population. But after everything was removed, we had a high resolution second run, and we removed a handful of clusters that were specific to a few individuals. And that left us with 27 clusters and about 325,000 nuclei at the end. And this is important to note because those previous papers that I talked about, the previous 4 papers that were in high impact journals, had fewer combined nuclei than this. And we can have a chat at some point about whether that money was worth it, because we didn't get a ton more nuclei or a ton more clusters out the back end, despite having many, many more times the nuclei. So there might be a law of limiting returns here. So what we did find after doing our clustering was, one, we used known cell markers to pull out the major cell types, and we did find all the major cell types we expected. So astrocytes, endothelial cells, inhibitory and excitatory neurons, microglia, oligodendrocytes, and OPCs. And on the left here, you can see that there doesn't appear to be any major difference in the proportion of any of these between cases and controls. However, what that's kind of hiding is that if you break it out on the right by subject, what you're seeing is I think one of the biggest benefits of single nuclei RNA-seq, and one of the problems with bulk lysate, which is that there's a huge variation across the population. So you can see one of these samples has a large number of purple, which means there's a lot of oligodendrocytes. And what that means is that the tissue chunk we pulled could have had more white matter than gray matter. And vice versa, if there's very little purple, maybe we pulled tons of gray matter and we're just getting lots of neuronal nuclei. And if that was a bulk lysate, those people would be hugely different probably in expression levels, but they wouldn't be that because of case control status. It would just be because of what sample you happen to pull the lysate from. So this allows us to get around that. You don't have to dig into this, and I think these slides will be up somewhere so you can look into it later, because this is a very messy way of presenting the data. But basically, we used known cellular markers, GAD1, GAD2, MAG, MOG, all those fun things to identify what our clusters actually were. So instead of just the major cell types, these are the full 27 clusters that we pulled out. A large number of neuronal subclusters, as expected, but also various types of glia also have multiple clusters. And I think the important takeaway from that is if you take a look at our entire dendrogram of how all of our cell clusters are related, and you just pull out the inhibitory neurons, we found stuff we expected to find. So this is sort of just a test of how valid is this? Are we getting what we expect? And what you're seeing here is just violin plots. So this is expression data for the different inhibitory neuron cell types. And what you're seeing is that we have a couple of cell clusters that have parvalbumin. We have a couple that have somatostatin. We have one that has VIP. And we have several that have varying amounts of CCK expression. So we're getting what we expect to get, which is multiple interneuron subtypes. I'm not showing it here because it's very messy, but the layer markers as well, we're getting certainly 2, 3, 4, 5, et cetera, excitatory neuron layers, layer markers showing up. So we got pretty much what we expected, but the alternative here is something I kind of mentioned before, is how many clusters is the correct number of clusters? And the answer is we have no idea. We found 27 clusters. The Nagy paper that did depression also found 27. And while we had roughly the same number of neuronal clusters, they found more OPC and oligodendrocyte clusters. We found more astrocyte clusters. So a lot of this depends on sample size. A lot of this is going to depend on just individual variation that you probably can't control for. A lot of these are still small sample sizes due to cost. And so it's not necessarily clear how many clusters of cell you should get in the end. And that's probably something we're going to have to figure out as the sample sizes grow, as the costs come down. So let's get into a little bit of the analysis here. Or rather, some of the issues that show up in single nuclei RNA-seq analysis. The biggest thing is that unlike bulk lysate, you're getting individual data points, a lot of individual data points for a single gene. And so what that means is that your gene that you're interested in is not going to be expressed in all nuclei from a cluster. So on the right here is just the first 20 genes that were expressed above 0 in one of our clusters. The x-axis is counts, and the y-axis is proportion. And what you're seeing is a bimodal distribution on all of them. There's this nice, presumably normally distributed curve that's off to the right, and that's the ones that are expressing it. And then you just got this giant peak at 0, and that's all the nuclei where your gene just didn't show up by chance. And so that's really been an issue that the field has had to deal with in a way that bulk RNA-seq really didn't have to deal with. And there's been people attempting to deal with this by creating new R packages or new programs to handle this. There's questionable whether those have actually worked. I think basically it's the Wild West is how I would put it. So first of all, of those 4 major papers I mentioned before, they all used different methods. So the Alzheimer's paper used a linear mixed model and a Wilcoxon rank sum test. Velmashev and Shermer were from the same lab. They did the MS and autism papers. They used MAST, and spoiler alert, we're going to use MAST in a couple slides here. And the Nagy paper used a different linear mixed model. So there's all these papers varied in the covariates they used, although they obviously had sex in there. Different FDR cutoffs, different log FC cutoffs. There really is no consensus yet in the way that, say, at GWAS, we have a cutoff that we just sort of universally accept as as appropriate, and we have sort of best practices on how to deal with this. This is still a field in flux. And I think that's basically this study from 2018, which showed that the analyzing data with programs specifically designed for single nuclei RNA-seq or single cell RNA-seq has no benefit generally over using anything else. So they took known data sets and they ran them through a bunch of different protocols you can see on the right here. Basically, stuff at the top worked better than the stuff at the bottom. But there's just a huge variation in efficiency. And there's been a number of programs, like I said, designed for single cell RNA-seq, and they don't necessarily do better than anything else. Like the sixth one down here is a t-test. A little bit below that is a Wilcoxon rank sum test, and then further down from that are programs that were literally designed from the ground up to analyze single cell RNA-seq data. So clearly there's been some issues with usability and efficiency of what's been designed so far. Also on here, just methods that were designed for bulk transcripts previously. Those have also been shown to generally be as good in a lot of cases. And I think the biggest takeaway here and what the authors really said was that a lot of this is going to be specific to the data set you're using, so you might have to play around and figure out which of these works best with what you have, which doesn't sound necessarily as scientific as we would like, but again, best practices aren't really nailed down just yet. We ended up using MAST with counts per million and detection rate, so it's the second one from the top here. Okay, so here's the hurdle model that we ended up using. The MAST hurdle model is designed around the idea that you've got this huge number of zeros. And so it's built on two models, a discrete model and a continuous model. The discrete model is the percentage of nuclei with a gene expressed different between cases and controls. The continuous model is when you actually do get expression, is there a difference in expression level? And hopefully these diagrams will help explain that a little better. So basically, how many times did you get non-zero numbers? And you're comparing cases control in the one model. And then the other model, you're removing the zeros, and then you're just comparing the means. And so the final statistic is basically just combining these two together. And that's your official hurdle model. So we did our differential expression analysis using MAST, like I said. The fixed effects in this case were case control status, sex, age, and batch. Normally batch would be like a random effect, but we only had 4 batches, so it doesn't really work out well as a random effect, so it was put in as a fixed. Gene detection rate is super important. If you're ever going to do this on your own time, definitely have to include this. The MAST paper, interestingly enough, is about half about how MAST is good and you should use it, and the other half is that no matter what you use, you should always have gene detection rate. And so gene detection rate is the number of genes that were detected in each individual nucleus. And it turns out that has an enormous effect on differential expression analysis if you don't account for it in the model. We also had a random effect of subject. And in this case, that's obviously because nuclei from an individual are more closely related to themselves than nuclei from other individuals, and so you have to account for that. And then finally, for our significance, we chose an FDR of 0.1 and a log FC of 0.14, which is a 10% difference. On the right here, you can just see a volcano plot. It's not super informative, but just to show that we have this nice spread, minus the one all the way on the right, STX-BP5L, which is ruining the symmetry of this nice volcano plot right now. But we did get a large number of differentially expressed genes. To be specific, we had 2,853 differentially expressed genes, which is 2,200 unique genes, because some of these were differentially expressed in multiple different clusters. The table on the right here is just a list of the different layers, the different cell clusters with the number of differentially expressed genes. And you can see here we had basically very top heavy. We had 96% of the differentially expressed genes we found were in these top 5 clusters, even though we did get differentially expressed genes in 21 of the 27 clusters. So one of the things that's going to show up later as a possible issue here is that these are all neurons. So we are very, very heavily neuronal with our results. And we're going to talk a little bit about why that might be the case and whether it makes sense. But our two big clusters here are a parvalbumin positive cluster, one of the two that we had, and then also a layer 5 HTR2C positive excitatory neuron cluster. So why neurons? I don't think I'm going to get a ton of pushback. Again, not a neuroscientist, but I don't think I'll get a ton of pushback on the idea that neurons are relevant to schizophrenia and important. But certainly there's been a wealth of literature on the role of glia, specifically in schizophrenia. So our results don't necessarily jive with those facts. However, a paper a couple years ago took the GWAS hits from the schizophrenia, the PGC schizophrenia GWAS, and they took those genes and they looked for enrichment based on single cell RNA-seq data from mouse brain specifically. And what they found was that the GWAS hits were enriched for genes that were expressed in mouse neurons. Now, this is from the entire mouse brain, so there's a couple types of neurons in here that we're not really going to capture. But cortical interneurons was certainly one of them. This is not obviously 100% correlation. It's interesting. Certainly GWAS hits aren't all genes that are relevant to schizophrenia, but this does give a little bit of credence to the fact that we found a lot of neuronal differential expression. And they did find similar results in limited human data sets. Furthermore, on top of that, again, parvalbumin positive interneurons have been looked at for quite a while in the context of schizophrenia, whether it's reduced density of those types of interneurons or altered gene expression of parvalbumin itself in schizophrenia. This is just a minor handful of citations here, but the list goes on. So certainly those are thought to be involved at some level in schizophrenia. A second question that sort of arises out of this after the neuronal stuff is, did you just get a correlation between clusters that had a lot of nuclei in it and differentially expressed genes? And the answer is no, actually. So you can see here on the x-axis is the number of nuclei in the clusters, and the y-axis is the differentially expressed genes, and we're just getting a smattering all over. There's no real correlation. This to me actually makes a lot of sense. This huge cluster up here is oligodendrocytes. In our case, we only got one cluster. It didn't cluster out, and when we attempted to cluster this alone at higher resolution, it just didn't work well. So this might be multiple different subtypes of oligodendrocytes lumped together, which again would ruin sort of your ability to find differential expression. Meanwhile, there is probably some minor power issue if you get too few nuclei in a cluster, but you're making up for that by having a very specific cluster, hopefully, which will get rid of a lot of the sort of the muddying the waters of having a mixed population. Another thing that we were really interested going into this was that we expected to find genes that were differentially expressed in one cell type, and you can see a couple on here on this heat map, but we were also interested in seeing genes that were differentially expressed across a wide variety of cell types. And so in this case, you can see that we do have some, downregulation. This is just a handful that were chosen for the heat map here. Some downregulation, some upregulation across just individual cell types. But then you'll see IL-7. IL-7 did not show up in any of our differential expression models for individual cell types. However, it appears to have sort of a broad downregulation across a wide variety of mostly excitatory neuron populations. And so I think this speaks to there's still some benefit possibly to bulk lysate, or there needs to be a little bit of difference in how you analyze the single nuclei RNA-seq down the road, because you've got to figure out sort of this pervasive downregulation. You might capture that in bulk RNA-seq and then miss that the way we're doing it right now, the single RNA data. So I'm going to chat for a bit here just about some things that caught our eye. Again, most of how you're going to analyze this is gene ontology and pathway analysis and stuff like that, because it's basically insane to go through this and look for genes of interest. But a couple popped out to us that I wanted to mention. Clock showed up, and after seeing clock on there, which is obviously a molecular clock circadian rhythm gene, we looked in and found there's actually a number that were significantly differentially expressed. So What you can see on the right here, the ones in this picture that have a red asterisk next to them are differentially expressed in the parvalbumin positive interneuron cluster that we were working with. And that's quite a number of this on just this bare bones molecular clock that we hit. This kind of makes sense. Schizophrenia, there's a long list of literature findings on sleep abnormalities in schizophrenia, and also more recently some attempts to look at the rhythmic expression of these underlying genes, and including one paper in 2016 that claimed Cry1 and PER2 just didn't have the same kind of rhythmic expression you're expecting to see in sort of non-schizophrenic individuals. The problem here, obviously, is that we don't have data on the time of death of these individuals, and so there is a chance that there's an underlying confound where, for example, people, the schizophrenia group just happened to die at specific times compared to the case group, and that could be confounding the whole thing. But still, pretty interesting and possibly relevant given the findings already in schizophrenia. Another one that showed up, and this is fun for me as a geneticist, we noticed that CACNA1C showed up. And for anyone who's not aware, on the right here is the PGC schizophrenia GWAS Manhattan plot. And I've pointed out where CACNA1C is. It's actually one of the most well-replicated findings genetically in psychiatric illnesses, other than probably some of the alcohol dehydrogenase, et cetera stuff. Also been associated with bipolar disorder. So very interesting that it showed up here, but there is some literature on schizophrenia as a channelopathy, meaning that alterations or disruptions in ion channels are part of the underlying pathology. And you see we hit a large number of calcium channel genes, potassium channel genes, and sodium channel genes. So possibly giving some credence to the fact that there's just some ion channel alterations going on in the background here in the cases versus controls. But like I said, I don't think going through and looking at individual genes is necessarily going to get us that far. You can't mine 2,200 genes, at least not in a short-term process. So we did start looking for gene ontology term enrichment. This is using the Panther database. And what we found is, if you run the two main clusters, we're probably going to run the top 5 eventually. I don't think we have any power below that, but for the purpose of this, I'm just showing the top 2. In the parvalbumin positive cluster, you hit ubiquitin-dependent protein catabolism, and I've just shown a figure from a paper on the right that shows that there's increased ubiquitinated protein levels in schizophrenia and also in treatment-resistant schizophrenia. So possibly interesting there. Spliceosomal stuff, like I said, we don't really capture spliceosomes. We don't really capture splicing events in the data set here, but there is definitely literature on splicing being relevant to schizophrenia. And then the big one here, and the one I'm going to talk about in the next couple slides, is mitochondrian. And there is a wealth of literature about hypotheses and data supporting mitochondria and energy issues and metabolism issues related specifically to schizophrenia. Which is interesting because ATP synthesis, again, popped up on the second one, the layer 5 neurons, but also vesicle fusion to plasma membrane, which could be related to neurotransmitter release, et cetera. So let's talk about mitochondria for a little bit. On the right here, you can see the electron transport chain, just a quick figure. And we hit things basically across this entire set of complexes. Most notably, and the one that sent us down this rabbit hole initially before we started doing pathway analysis, was the NDUFV1, which has been previously linked to schizophrenia, but was downregulated in 5 different clusters. And when we started digging into this, what we found was just pervasive downregulation of various members of complex 1, complex 3, complex 4, and then at the bottom here, MTFR1, which is mitochondrial fission regulator 1. There's only 4 of these. The ones that are bold and underlined are upregulated. Everything else is downregulated. So there appears to be specifically something happening here with mitochondrial genes that are encoded in the nuclear genome. So we started doing Ingenuity pathway analysis. Ben has been working on this recently, and this is very, very ongoing. I got this picture within the last 48 hours. But this confirms what we already suspected, which is that based on the Ingenuity analysis, everything in green, which is complex 1, 3, 4, and then the ATP synthase complex 5 at the end here, all of those are expected to have reduced function in the parvalbumin positive interneuron cluster based on the number of genes that are downregulated in those complexes. So very interesting. I don't have any real answers for why this cluster in particular versus other stuff, and perhaps someone with more neuroscience background might be able to make an attempt on why that makes sense. But yes, very, very interesting, that just very pervasive downregulation across this entire thing. And then the one thing that's ongoing right now is transcription factor analysis, and this is being done by Andrew Weller, who's a postdoc in Wade Berrettini's lab, and also by Glenn Doyle, who's a senior researcher in the group. And what we noticed was that there's a number of transcription factors that show up as differentially regulated in our data set. And so you can start to make hypotheses about, okay, if this transcription factor is differentially regulated, are you seeing downstream targets also differentially regulated? And you can form sort of what's the proximal and what's the most distal relevant thing. If you're going to target something, should you be targeting the transcription factor and not the downstream differential regulation? And so they are identifying target genes using TF2 DNA. And they're doing enrichment analyzing with David. And this is just one quick example of what they found, because again, they are literally doing this right now. And so we had downregulation of ZNF44 in one of the clusters. And it turned out there were a number of genes that were known targets of ZNF44. 4 of them downregulated, 1 upregulated that you see in red here. But all of these have GO-term cellular compartments for synapse. So you can start to sort of tease apart for each of these clusters, are there relevant transcription factors that we should be targeting that may explain a large amount of the differentially expressed genes downstream. So I think it's important now to have a discussion, because otherwise Wade will bring it up, because he brought this up initially, that where have all the glia gone? So if you look at this picture on the left here, that's our breakdown of the major cell types, and you see about 55-ish% excitatory and inhibitory neurons combined. And then on the right here, you can see from the Alzheimer's paper that was Mathis et al., they had 63% neuronal. I don't have some good pictures for the other ones, but they're very similar. The Nagy paper is even more skewed, probably like 70-30. And the problem is that if you look at data on how this ratio should be, it should be flipped. There should be more glia showing up here than neurons. And so here's just a thing from Aceveda in 2009. They estimated that in gray matter, cortical gray matter, it was 1.48, so 48% more glia than neurons. So this fits with actually some data we did for a separate project where data from some of these cases and controls were FACS sorted to get us neuronal nuclei. And that one had a higher proportion of glia than neuronal nuclei. So the question is, this does not appear just to be us. It appears to be all the other 10X papers. Where have all the glia gone, and are we missing something very important? And the answer is, where have they gone? Not clear. It could be the nuclear isolation. Certainly there's portions in there where if there were density issues, Glenn Doyle from our lab brought this up initially. If there were density issues, maybe you just lost some during the action, some of the spins. I'm more partial to a UMI issue. And so I haven't shown you on here, but the nuclei from neurons have much higher UMI numbers than the glia, going all the way down to our lowest cluster, which was the microglia cluster. And so we are making cutoffs to try to get rid of the noise at the very low end, because those are really uninformative cells. But if there's just a pervasive low number of UMIs, which would correspond to just low numbers of transcripts from those nuclei, you might be cutting out useful glia. And if you're trying to figure out if that's bad or not, I don't know the answer to that either. It could go either way. If we're just taking a haircut on just pervasively across the board, we're losing X% of glia from each cluster. That's not actually that bad. You can force your way past that just by increasing sample size, increasing the nuclei you capture. My bigger worry is that we're losing specific glial clusters that just have low UMIs, and so we can't differentiate them from the noise. So I think this is something that is going to have to be figured out by the field, and whether it's bioinformatic in nature, whether it's technical in nature, someone's got to figure out, one, does this make sense? And if it doesn't make sense, like what have we lost and how important is it? So let me just wrap up with what our future directions are going to be here. I think an important thing to note here is that of those 4 major papers, RNAseq was the way they chose to validate a handful of the differential expression events. It's also probably worth noting that it appears that none of these actually had that on first submission. They were told to go back and validate some. And you can see this clearly as some of them are very stitched on at the very end. But we happen to be very, very close in our building to someone who knows how to do RNAseq, which is Dr. Lauren Stein. She's a postdoc in Matt Hayes' lab. RNAseq, for anyone who doesn't know, there's a cartoon on the right here. Basically, it's fish with a couple of caveats here. So you've got these Z-shaped probes, and they bind next to each other. And so when they bind next to each other on the target mRNA, you get this whole scaffolding building, and attached to that are fluorescent molecules, and you can look at up to 3 genes simultaneously on a slide. And so what we're going to do is hopefully in the next month or so, Lauren is going to go through. She's going to take 5 of our samples, cases, 5 controls. We're going to blind her, and she's going to run through some of our genes, trying to validate our differential expression findings. Also going forward, I think replication is going to be a huge deal here. Like I said before, the major papers and also our paper our study have 25 to 48-ish samples. And I think what you're going to find is that, yes, it's cost prohibitive, but as you get more and more people, you're going to have a much better sense of what the clusters you're expecting are. The individual variation is so big when you only have 16 cases and 16 controls that it's just going to pull things in weird directions. You're going to end up with people separating the clusters that may not be real, or vice versa. It just might be so diverse that you're not seeing a split out of 2 clusters. They're just being merged into one. And again, you're losing some of that granularity and specificity. And we also put an R01 application recently, hopefully to do 32 and 32 samples soon. And the other one here is for everyone who knows me, I don't work on schizophrenia normally. I work on substance use disorders, specifically genetics and pharmacogenetics of opioid use disorder. And I think no one's done it yet, but I think this technique is going to be very, very helpful in teasing apart some of the underlying biology for substance use disorders. NIDA put out a program announcement looking at, for animal models in opioid use, specifically looking at single cell techniques. And I think you can imagine why, because you can set up mice for self-administration or rats for self-administration, and you can do passive versus active. You can do before and after, because you've got mice that are genetically identical. You can do extinction paradigms. You can do all sorts of stuff that will tease apart the individual parts of opioid use, despite the fact that mice obviously can't get opioid use disorder specifically. But also this lets you bring to bear just an enormous amount of genetic information that we have on different mouse strains, high taking versus low taking. There's an amazing amount of work that could be done here. In addition, opioid use disorder, we actually have a collaboration with Deborah Mash down in Florida. And so she has cocaine use disorder and opioid use disorder brains from people who died of overdoses. And so we have an R21 that's being reviewed possibly tomorrow that we're hoping will let us do a small pilot study on that to look at opioid use disorder versus controls. So that's all I've got for you. Obviously, the acknowledgements start with Ben here, because Ben and I have basically been co-conspirators in this whole thing. But also thanking the entire Berrettini lab. Wade funded most of this on this R01 here. You can see in the bottom right. thanking the Hayes Lab, specifically Lauren, Rachel Kember, who's going to help us with some analysis / making pictures that are publish worthy, because my experience mostly is line graphs and tables, and that's not going to cut it for this kind of thing. And then finally, the Center for Applied Genomics, they did great work for us. So with that, thanks for having me, and I will take any questions.
Thank you, Rick, so much. I have it's Chuck O'Brien. I have a question about, early on in your talk, you described in a lot of detail the separation in the neurons, and you classified them as excitatory or inhibitory. And I was wondering if you could explain how you make that classification, because an excitatory neuron can, depending on where it synapses, can actually cause functional excitation. And in some, especially when you get into the opioid system, they cause modulation. In other words, can be varied according to the number of inputs from which category they come. And so I'm not sure what you mean by an excitatory neuron, for example.
Yeah, I mean, so the truth of the matter is I don't know either. We're stealing lingo that's been used by people before us, the Blue Lake paper, the Mathis paper. We have gotten that exact comment, or rather people in our group have made that exact comment about the sort of vagaries of excitatory versus inhibitory and what we should actually be calling them. I don't have a better term for them, but I am all ears for something that would make more sense to people in the field. But yeah, we're just basically cribbing these from people who already published.
Yeah, it might be helpful to put it into the context of work coming out of the Connectome projects. I realize you don't have any circuits in the models you have because they're frozen and they're
right.
all juice at that point. but it's as we're learning about the brain, it's it has to be put into context, and somehow the genetics have to be integrated with the circuitry.
I absolutely agree.
Rick, I think this was extremely elegant. It's also really daunting for me to think about this granular level, which is probably where a lot of the mysteries reside, and at the same time, the big meta data approaches that, basically big data, they're complementary, but wow, there's a lot of things to look at. So whether you get thousands and thousands of individuals or thousands and thousands of genes clusters and now across anatomical regions, it feels, cell types, it begins to feel like a lot. And so I'm thinking of you talking from the mountaintop. What does the landscape look to you? Is there going to be a natural tension between these big data approaches and these very, very single cell approaches? And will there be, a happy cohabitation? Is there, what are your thoughts?
Well, yes, it's a problem. Absolutely. So yeah, like you just explained one of probably the biggest problem, which is, okay, let's, for the sake of argument, say that we figure this out down the road of DLPFC, schizophrenia versus control. We're pretty confident about that. It's like, okay, well, you can't just port that over to some other part of the brain. And now it turns out there's lots of different parts of the brain. And so the amount of data you would generate is almost inconceivable just for like a single human brain.
It's hard to put your head around it or your hands around it. And I mean, it's admirable to be able to have these granular approaches, which probably do, obviously, probably lead to unveiling some of the mysteries and eventually resolving things. But they're sort of multiplying the level of the data and levels of the data and the observations by several fold that were already very high in terms of just the sheer number of genes, even from a bulk approach. So while it's very likely to be the quote-unquote right thing where a lot of the individual variation lies and where some of the answers lie, wow, it's a lot.
Yeah, I think you're looking at something in the form of, deep learning, AI, machine learning type processes where you're just going to have to take an absurd amount of data and dump it on a system and let it try to figure out what's important, because it's going to, like, even addressing 2,200 genes and trying to figure out what it all means is not particularly easy for a group of individuals. Once you're into multiple brain regions and you've got hundreds of people for each brain, it's just going to spiral out of control.
Right.
So it's taking it to a translational level is going to be
challenging.
Interesting.
Do you see yourself going in this single cell direction with the work that you're going to be doing? I know that you'd start to do genome-wide work, and so these two things are complementary. What do you see for yourself?
Well, I mean, for the time being, we've put in a number of grants using this method. I think, one, because it's timely, and there's just not that many people who are doing it or have evidence that they've done it. So,
Right.
if you've got a hammer, everything looks like a nail. So I think this is going to end up being complementary down the road. But for right now, we're trying to get the 10x stuff just because it's timely. I think there is definitely a place to integrate genetics, like I said, especially with the mouse models and stuff like that, integrating genetics in with this kind of 10x data, so you can see why, try to understand what these polymorphisms are actually doing will be very, very useful. Whether we get there with humans anytime soon, probably questionable.
But still an elegant technique, and I do feel a member of the great unwashed in terms of the elegance of these techniques. But I appreciate you walking us through it and giving a very obviously careful and detailed analysis. So for those non-geneticists in the audience, take heart. The field is in good hands.
Thank you.
Anybody have any other questions? We have about another minute.
I just wanted to commend Rick for giving exposition and detail about this new method. And there are important caveats as we try to study opioid use disorder. As you can imagine, between the schizophrenia cases and controls, there may be a host of differentially expressed genes that are the result of long-term antipsychotic treatment in the cases, which is almost entirely absent in the controls. And so you can extrapolate to the case of opioid use disorder, and there we would be dealing with differentially expressed genes that may be due to chronic opioid exposure, which again would largely be absent in the controls. So there's a lot of groundwork to be done.
I think there's a mountain to climb there, Wade. But you've got good company, you guys. And a good team. Thank you so much for taking us through this. And as you get your GWAS studies going, we'll be happy to hear about those too. I know you're going to talk about delta receptor allele stuff at some point, so we'll look forward to an encore performance. Thank you very much, Rick.
Thanks for having me.